to be here on a Monday after break, so thanks for coming. So we're going to start right where you guys left off last time, talking about the structure of vesicle release. So I know that Dr. Etheridge introduced this slide a little bit, but we're going to go ahead and reintroduce it because I know it's been a week, so feel free to ask questions as we go, because I know that this is a lot. But this part down here is actually something that is not really important for our purposes, so this isn't something that I'm going to ask you guys to know. The main stuff that we're going to be focusing on is particularly on the left side of the screen up here. And so what you see is you have your vesicle in gray, and this little complex right here is called your snare complex, so we're going to talk about that in detail, specifically about how this complex sort of helps stick the vesicle into the membrane so we can get that neurotransmitter release. We're also going to talk a little bit about these proteins over here, which we will refer to as scaffolding proteins. We'll talk a little bit about each one and what they do. So just to orient you in general for what we're seeing here. So this, of course, is a voltage-gated calcium channel. So you have what you can see is calcium coming in and calcium is binding, and that binding of calcium is what's going to initiate this whole system of transmitter release. So jumping right in, there are several components that are really important for us to know, and the best way to divide them in terms of understanding is by where they're attaching. And so it's unfortunate that a lot of these things have very, very similar names, and so it's important for you guys as we're going through to try to associate each name with not only what it does, but where it is sort of in the whole process of neurotransmitter release. And so the first thing that we're going to talk about are the portions of, or the molecules and the proteins that are attached on the vesicle. And so the first one that we're going to talk about is synapsin. So do you guys remember talking a little bit about axonal transport, where you would have your actin filaments are sort of like your railroad tracks in your axon, and down those actin filaments you can have things trafficked that need to go to the synapse. So this is your axon right here. You have your little actin filament train tracks, and it's down these filaments that your vesicles are going to come after they've been recycled, right? And so you have your vesicles trafficking down the axon, and Dr. Etheridge sort of left off with them reaching this point. So what happens when they actually get there, when they get to the synapse, what happens next? And so what you see is that you have what we call a reserve pool. So the vesicles actually sort of bunch up here at the top like little grapes, right? So they're still attached to actin. So when you think of a bunch of grapes, they have their little stems, right? So they're hanging from the top of the synapse up here by the actin, right? And what is attaching them to the actin still is a protein called synapsin. And so, you know when you pull a grape off the stem, how there's sort of like a little knob on the end of the stem? That's like what the synapsin molecule would be when you're thinking about the grape analogy. And so synapsin is important for sort of keeping these vesicles there at the top until they're needed, right? Remember, this is our reserve pool. And so on synapsin, what you have is a binding site for I always pronounce it wrong, calcium calmodin kinase, right? So we talked about calcium calmodin kinase 2 in a different chapter. Now we're referring to calcium calmodin kinase number 1, right? And so the important thing about calcium calmodin kinase, or KMK, we're just going to call it calcium calmodin, the important piece of information for this is it's calcium dependent. And so this is the first place that we're seeing calcium interacting with something in this system. And so calcium comes in and it attaches to calcium calmodin and it sort of activates it. And then from there, the calcium calmodin comes in and actually phosphorylates the synapsin. And so this phosphorylation by calcium calmodin actually allows these vesicles to detach and drop down. So this is your active zone down here, and after calcium calmodin comes in, you can get movement from this reserve pool down to the active zone where you're going to eventually have neurotransmitter release. So remember, this is sort of step 1 in this whole system. And one thing that's important to remember here is that you don't necessarily always need these to drop from the reserve pool. So as we get further down, we're going to talk about how you're actually going to have vesicles that are already docked down here and ready to go. And so this is just one area in which calcium is going to be important. And so what we're going to talk about as we go through is that when calcium's coming in, so let's go back to this image right here, we can see that this is the membrane, right? So the voltage-gated calcium channels are going to be down here on the bottom of the active zone, right? And so if you actually have vesicles down here, it's going to take quite a lot of calcium to get all the way up there to activate the synapsin, right? So in what type of instance do you think that you would see enough calcium to activate this reserve pool? So we're using voltage-gated calcium channels. So what type of situation do you think you would get enough calcium to actually have vesicle release from here? I know it's right after spring break and we're all still kind of getting back into things, but a guess is good too. So they're voltage-gated. It's a pretty good hint, right? So what do you need to get activation of a voltage-gated channel? Just in general, what has to happen? Okay, I hear it like more. Keep going. That was good. Louder. Anyone? I heard it. Somebody had it. Right, so you're going to have some sort of voltage change, right? And so to get enough of a voltage change to have enough calcium coming in to reach all the way up here, you're going to have a pretty significant change in voltage, right? This is like either, you're going to get several action potentials in a row, some sort of situation in which you're going to need a lot of neurotransmitter release. And so as we go through, what we're going to talk about is you don't always have mobilization from the reserve pool. So sometimes you're just going to get enough calcium coming in to activate neurotransmitter release from down here. But if there's a situation in which you need a lot of neurotransmitter release, right, you're going to get several action potentials that are essentially going to have enough calcium coming in to have vesicles pull down from this reserve pool. Does that make sense? All right, excellent. Okay. It's my, All right. So next on the vesicle, we have the V snares. So for your snare complex that we talked about from that first picture, this is your snare complex right here. You have your V snares. They're on the vesicle, V snares, vesicle. And then you have your T snares. So your T snares, T stands for target. So your target is the cell membrane. That one's not quite as easy to remember. But the V snares one is good because you can remember V and vesicle. And so there are a couple different kinds of V snares that we're going to be talking about. The first, and very important, is synaptobrevin. And so your book also refers to it as VAMP. So for our purposes, we're going to talk about synaptobrevin. All of the images that we're going to look at today are going to use synaptobrevin instead of VAMP, but if you're reading and you see VAMP, those terms are used interchangeably. And so synaptobrevin, you can see here, it's sort of shaped, sort of like, it has like a little knob on the end almost, sort of like this. And so what you see is that this little knob is actually what's going to interact with the snare complex. And so it's attached in here through the vesicle, so it's sort of anchoring the vesicle to this complex when they attach. And synaptobrevin's job is to bind to the T snares when they come in contact. And so one of the things that it helps to consider is that when these drop down from the reserve pool, when your vesicles drop down, they're not like, so let's say you have a T snare right here, they're not just dropping down and making a beeline for that T snare. We'll talk about proteins that are in place to sort of funnel them into the right direction a little bit later. But in general, for the most part, they drop down and they're sort of like floating willy-nilly around the synapse. And nothing's going to happen until they bump into a T snare. So, like I said, there are proteins that will sort of help funnel them in the right direction. But in general, they're floating downward, but they're not making any sort of directed movement towards the T snares. So, obviously, if you have a lot of them all pushing downward at once, it's going to make it easier for them to hit a T snare. But for the most part, remember that while they're floating down, they're not necessarily actually making sort of active movement towards those snares. So when they bind is essentially just when they contact each other in the synapse. And so the binding of the V snare to the T snare is going to help pull the vesicle down into the membrane. Does that make sense? right, so another part of the V snare is synaptotagmin. And so synaptotagmin is really cool because it is sort of one of the most essential pieces for neurotransmitter release. So without synaptotagmin, you're not actually, you might get, things might dock, you might get the vesicle going to the membrane, but you're not actually going to get that fusion of the membrane and that release of neurotransmitter. And so synaptotagmin is crucial for binding calcium. And so I want to highlight here that this does say 5 calcium ions. And so there are instances, there are certain neurons that actually, depending on their structural capabilities, will bind more or less. But a general rule of thumb is that synaptotagmin is going to bind 5. And so that's the number that we're going to go with for this. And so what you see with synaptotagmin is that it actually has 2 different binding sites. So it's CB1 and CB2. So calcium binding 1, calcium binding 2. And so the first calcium binding site is going to bind 3 calcium ions. And the second binding site, obviously by definition, is going to bind 2. And so these 2 different binding sites sort of help synaptotagmin perform its 2 major functions. So the first function is that calcium binding essentially allows synaptotagmin to sort of turn on. And so it's going to create a slight structural change in synaptotagmin that's going to, one, make it more attracted to the membrane, so it's going to sort of pull it down into the cell membrane. And it's also going to cause it to shift in to this snare complex, and we'll talk about this more in detail later. It's going to bump this little complexin molecule out of the way, which is going to end up being really important for allowing that release of neurotransmitter. And so the second binding site, this binding site 2, the binding of those 2 calcium ions allows synaptotagmin to fuse with the membrane, which is going to provide the pressure necessary to sort of pull that membrane open and apart. And so we can go ahead and move over, and we'll talk about it all through for a picture because I know that that's a lot. So over here, you have your vesicle that hasn't been docked to anything, right? So you have your synaptotagmin here, your synaptobrevin is up here, and then here is these are your T snares, right? These are your snares that are attached to the membrane. And so one thing I think that is important to notice is that these are sort of in an upright position, right? And so you can see that when these bind, they actually compress down together, right? So what you have is your vesicle moving down, and your synaptobrevin is attaching to your T snares. We're going to talk more about the T snares later in each individual piece, but you see your synaptobrevin is attaching to the T snares, and that attachment actually allows this structure to condense down to pull the vesicle closer to the membrane. And then after calcium comes in, calcium binding allows the synaptotagmin to swing in. It knocks that complexin molecule out of the way, and then you essentially have enough tension necessary to pull the vesicle open and release the neurotransmitter. Is everybody good so far? Okay, we're going to talk about the T snares, and we're going to go through essentially this all again a couple more times. So if by the end it's still not making sense, that's a good time to flag me and ask questions. So now let's talk about the pieces that are on the cell membrane, right? So remember, our V snares are attached to the vesicle. T snares, our targets, are on the cell membrane. And so the 2 that we're going to talk about are syntaxin, and SNAP25. And so you don't necessarily need to know the difference between these 2. Just know that syntaxin and SNAP25 are T snares. And so obviously a good way to remember this, syntaxin has a T, T snares have a T, and there's not a great way for SNAP25 other than if you spell out 25, it has a T too. So syntaxin, SNAP25 are your T snares. And so again, these are responsible for binding to your V snares, so your synaptobrevin specifically, and pulling that snare complex down together into the membrane. And so the way that you get that structural change that we noted in the last image is that when the T snares and the V snares bind together to make the snare complex, there's a release of energy that's needed to create that structural change and sort of condense everything down closer to the membrane. So it makes a lot of sense that if you're going to release neurotransmitter, if you have to fuse into the membrane, you want to be as close as possible. And so the added energy that comes from those two things binding together is part of what helps condense that down and pull the vesicle as close to the membrane as possible. Make sense? All right. Okay, so there are other proteins that are responsible or are associated with neurotransmitter release, and we're going to talk about these in terms of scaffolding proteins. So you have first your ribs and beams. Examples are RIM and MUNC13. We're going to talk a little bit more about those as we go through. But the important thing to remember for the scaffolding structures is they essentially work the same way in a biological system as they do like in architecture, for instance. So if you're providing a scaffold for a building of some type, you're essentially providing that structure needed to serve some sort of purpose. And so for our case, these scaffolding proteins are responsible for holding the vesicle in place where we want it to be, or in some cases they're responsible for funneling the vesicle down so it can be docked properly in the right space. And so you have your RIM and your RM here, the RIMs. MUNC13 and RAB are all scaffolding proteins. So your RAB protein, that was something that you guys talked about in relation to axonal transport. And essentially its main function here is that same sort of idea, like where it attaches things to actin in axonal transport. Here its only purpose is to attach the vesicle to other things, basically. And so in this case, it's going to bind to MUNC13, and it's going to provide sort of some extra stability to keep the vesicle where we want the vesicle. And so MUNC13 is part of a class of MUNC proteins. So MUNC actually stands for mammalian uncoordinated proteins. And so there's a ton of different MUNC proteins. There's actually a whole group of them. They're described as uncoordinated because they're essentially responsible for providing scaffolding for the snare complex across a wide range of, so cognitive function, but where you see the most impact of MUNC proteins. So if we were to disrupt your MUNC system, you would see actually disruptions in movement and coordination, and so that's why it's called mammalian uncoordinated. So if in a rat you knock out their MUNC protein systems, they have trouble walking and balancing. And so, which makes a lot of sense when you consider structure, right? You need to have the correct structure to have that good balance, right? That's a stretch, but hopefully that will help you remember. So, there is another MUNC protein that we're going to be talking about, MUNC18. And so really the only thing that's important for understanding about these different proteins, the MUNC proteins in particular, is that MUNC13 is associated with scaffolding, and MUNC18 is associated with the snare complex. So we're going to talk about that in just a few minutes. So in terms of what you need to know from this area of the slide is generally know the purpose of scaffolding proteins and be able to distinguish between which MUNC is responsible for which job. So which MUNC is a scaffolding protein and which MUNC is involved in the snare complex. Does that make sense? All right, perfect. Interestingly enough, while you don't see as many naming conventions here, there are a whole mass of scaffolding proteins that are associated with this that are actually named by the parts of a boat. I'm not really sure who thought that was a good idea, but there's a whole system, like you have the topmast, you have the bow, and you guys don't need to know any of these. I think there actually was a slide in here at one point for those, but you don't need to know them, so it's not in there anymore. But the idea is that these proteins are sort of there to provide the same sort of structure as a mast would on a ship. So, all right, so some of the other players in this system are MUNC18. So again, this protein is involved in the snare complex. It's this cool little boomerang shaped guy right here. And so MUNC18 is really just responsible for sort of stabilizing that whole complex. So you can almost think of MUNC18 as like a safety pin. And so after the complex is all bound together, so you have your T snares and your V snares that are attached together, MUNC18 can come in and sort of pin the whole complex together to keep it stabilized and keep it from detaching. And so MUNC18 you will also see will on occasion bind to syntaxin, and by binding to syntaxin, it actually makes it more likely for syntaxin to bind to your V snares. So in general, MUNC18 both helps make this complex form together, by making syntaxin more ready to bind, you might say. And then it keeps it in the right position by sort of pinning through the middle. Right. Make sense? All right, this final player that we're going to talk about is incredibly important for this system. So complexin is this little molecule that we saw right here. And so you can think of complexin almost as like the zipper portion of this T-snare complex. And so complexin is sort of put on top of the T-snare to keep it in the right position. And so complexin actually does its job so well that if complexin is attached to the snare complex, you're not going to get a release of neurotransmitter. And so what you need to happen, we're going to go back a few slides here, what you need is tension put on these different complexes to pull the neurotransmitter open to get to pull the vesicle open to get that neurotransmitter release. And when complexin is attached, it's stabilized in a position that doesn't give the tension necessary to pull that vesicle open. And so what has to happen is complexin has to be removed. And so this is where synaptotagmin comes in, right? And so complexin is bound to the molecule. Synaptotagmin actually swings in and knocks complexin out. And that actually destabilizes the snare complex enough that you're going to get the tension necessary to pull the vesicle apart and get release of the neurotransmitter. So, you guys feel okay? Okay, so I like this image because it shows really clearly the different components that are associated with this. And so you have here in yellow is your monk 18, yellow and brown right here. Your complexin is in pink. You have your snap 25 up there in green. And what you see happening is that when this complex binds together, they're sort of intertwined, right? Which keeps things sort of in place necessary to pull apart after complexin moves. But this is where calcium comes in and where calcium is very important. If you don't have calcium coming in, you're not going to get the movement of synaptotagmin, and then you're not going to get the pushing of complexin out of the way necessary to get the movement of the vesicle. Does that make sense? Okay, so one excellent way, we're going to go from the top again to remember this, but one thing I think that is helpful for remembering the role of synaptotagmin is when calcium comes in, synaptotagmin is essentially tagging out complexin, right? So it's saying tag, you're it, which means that the vesicle is ready to open and release the neurotransmitter. And so, let me see what this is. Yeah, so from the top, as we go through for this example, we're going to make a distinction between priming and super priming. So this isn't incredibly important for you guys. Like there's not going to be some sort of question like what is priming and what is super priming. The idea here is that you're getting the vesicle docked when you're primed and ready to go, and when you're super primed or like extra ready to release neurotransmitter, it means that you're moving complexin out of the way, right? And that is sort of the stage at which you're absolutely ready to release neurotransmitter. And so from the top, again, we have our transmitter coming down. We can see our synaptobrevin and our synaptotagmin that are attached to the vesicle. So again, these are your V snares. On the bottom here, you have your target snares, so your T snares with your syntaxin and your snap 25. And so as the vesicle drops down from the reserve pool, when synaptobrevin makes contact with your T snare, you're going to get a binding here. And that binding, again, has enough energy, or it releases enough energy to allow a structural change that then pulls that vesicle in even closer to the membrane. And so Here, at this point, you would call this, this is your primed position, right? It's docked and it's ready to go, but nothing's actually gonna happen until calcium comes in. And so, I know this is asking you to remember like a week and a half ago, but Dr. Etheridge talked about in the beginning that we knew that calcium was important for triggering neurotransmitter release, but we weren't really sure how. And so what she mentioned was that if you add calcium to a system like this, you're gonna see release of neurotransmitter within 1 to 2 milliseconds, right? So that is really, really fast. And so one of the main questions that researchers had was, what system is in place to allow for release that is that quick, right? So if we had to start from the top where we're releasing vesicles from the reserve pool and they're coming all the way down, it would certainly take more than 1 to 2 milliseconds, right? And so this sort of docking priming position is what allows for that very, very quick release of neurotransmitter. And so calcium's gonna come in, it's gonna bind 3 to that first binding site on synaptotagmin, which is going to allow for that swinging in and the knocking off of complexin. And then you're gonna have 2 bind to that second binding site, which is going to allow the synaptotagmin to fuse with the membrane, right? So it doesn't matter if you pull the membrane open, right? If you pull the vesicle open, if you're pulling it open inside the cell, right? The point is to release the neurotransmitter outside of the cell. So that's part of what makes that second binding site so important, is you're in position, you have the tension needed to pull the vesicle open. After the synaptotagmin allows us to fuse with the membrane, then you can get that tension to pull open and release the neurotransmitter into the synaptic cleft. Does that make sense? So, if there's a huge release of calcium, so let's imagine we have our voltage-gated calcium channel coming in there. If you have a large release of calcium necessary, or that sort of if you fully bind all of the calcium receptors down here, and you're still getting an influx of calcium, that's where you're gonna get the release from your reserve pool, right? And so one thing that's important to note is the max amount of calcium that you need to get release of neurotransmitter is 5, right? So what you can see on this image is that there's actually, so you have a snare complex here and a synaptotagmin here, and you have a snare complex here and a synaptotagmin here. So it helps if you're getting pulling from both sides, but you actually only need activation of one side to get release of neurotransmitter. And so, even if you only have 5 calcium ions coming in, if they all bind to one synaptotagmin, you're gonna get release of neurotransmitter. And so the more calcium influx that you have, the more neurotransmitter release that you're gonna get. Does that make sense? All right. Is there any questions about any of that so far? I know that's a lot. So the best way, I think, to practice this is write out the steps. So I know that it takes a lot, but if you can get the order down and where pieces go in the order, you guys are going to be good to go for the exam. Makes sense? All right. So this, again, looks like a lot. This is really just to give you guys an idea of one of like the life cycle of a vesicle and sort of how this all looks in action, you might say. And so up here, I like this image because it has the vesicle sort of blown up where you can see. So we have our synaptotagmin attached, we have our synapsin and our synaptobrevin, and then our RAB3, which remember is one of our scaffolding proteins that helps it attach correctly. And so in here you can see that you have a vesicle that's fused with the membrane and it's releasing its neurotransmitter. And then over here you have clathrin, right? You guys talked about that last 2 weeks ago, I guess now. You have the clathrin that's binding and sort of endocytosing new vesicles back into the membrane to be recycled. And so up here, again, these vesicles have their own pores and ion channels necessary to fill with neurotransmitter. And so that's what you see going on right here, right? Is you have acetylcholine that's going into this and refilling this vesicle with the neurotransmitter that it's going to need for later release. And then they're back up here at the top for storage, right? And so if at some point, let's say you dropped your reserve pool down, you're getting active recycling of these vesicles throughout this system. But let's say you still have a huge influx of calcium, right? As these vesicles are created, they might actually go right back down into mobilization to be ready to be docked. So instead of, they might sort of skip that step of coming up to be attached for reserve. But let's say our calcium influx is over, they're gonna then pop up at the top and be stored with actin and synapsin until they're ready to be mobilized again. Does that make sense? All right. Okay, so is there any questions? So that's actually it for today, guys. So if you have questions, it's a good time. Yeah.
On the previous picture before that.
This one?
Yeah. That one. So you were saying that middle one right there is
This one?
priming, right?
Yes, this is primed.
So
Yeah.
where would be like super primed?
Super primed.
One and the next one?
So it would be like if this weren't fully open yet. So after you've knocked complexin out of the way, that's your super prime.
Oh.
Yep, that's a good question. And like I said, like it's great to clarify to sort of, I think it helps understand like the minutia of the process, but there's not going to be an instance in which I'm going to ask you to clarify or to understand which is priming and super priming. The main thing to get from that idea is that you're primed when you're attached and ready to release, but you're not releasing it, right? That priming is what allows for such a quick response for neurotransmitter release. It's a good question. Anyone else? Like 45 minutes on my first lecture. It's not so bad. I'm letting you out early, so